Now, I think there's also abstracts we've done. We've been able to enjoy the collaboration with Kathy Wu and her group and the Broad Institute and with Dr. Getz. And there's been a tremendous amount of improvements in next generation sequencing. And we're doing work as well where you can actually analyze the transcriptome, or the genes that are expressed in a single cell. It's quite amazing. And you can characterize that cell, so we can now get a full composite expression of the genes that are expressed. You can also do a deep dive and look at the genes that are there, what mutations are there, what changes might be occurring. And a problem that's always been on the minds of people who treat patients with CLL is this issue of Richter's transformation. A very devastating outcome. It could happen without prior therapy. It can happen after therapy. And it's also, I think, something that is of great concern because the outcome is very, very poor. I'm hoping we can change this, and I think we can. In other words, we can probably find agents that make Richter's transformation a much easier disease to treat. I'm confident of that. So, but understanding is very important. So the study was to look at cases of Richter's transformations, and actually through multi-institutional work, a large collection of cell samples from patients with Richter's transformation was gathered along with their CLL cells prior to transformation. And then there was also patients who had Richter's transformation cells that could be analyzed. And so it was a deep dive into the genes that were expressed, the genes that are mutated, and to look at what factors might be associated with the two types of Richter's transformations. In other words, Richter's transformation comes in two flavors. One flavor is out of the blue, a patient with CLL is just minding his own business or her business, and all of a sudden develops a diffuse large cell lymphoma. Well, it happens that sometimes lightning can strike twice, and you can develop another cancer on top of the leukemia. And so there are cases of Richter's transformation which are not clonally related to the leukemia cell. And this has a different biology than those cancers that derive themselves from the leukemia cell. So one Richter's transformation is to have mutations that are accumulating within the leukemic cell over time, and then one is allowing that leukemic cell to trip the line and become an indolent disease such as CLL, and then trip over and become a diffuse large cell lymphoma. Now, these diseases look very similar. Namely, you look at the cells under a microscope, pathologists will say this looks like diffuse large cell lymphoma, and it shares many characteristics. So these two flavors of CLL are very distinct only in their origins. Namely, you have to trace back to where this leukemic cell, where this lymphoma cell came from. And you can do that by looking at the genes that are used to make immunoglobulin. In the case of clonally related Richter's transformation, you find that they may share a similar gene rearrangement that is a signature for that. In the case where they do not have similar gene rearrangements, then you can assume that maybe it's a de novo. Now, with the more recent study that's going to be presented at the ASH, we have further actual evidence that that is indeed the case. Because if you really do a deep dive into the genetics, you can find very peculiar constellation mutations. And if you look at these cases that are derived from the CLL and compare that with the cases that are not derived, they're occurring independent of the clone, they're different. And those other cases are more similar to the de novo spontaneous diffuse large cells that may occur in patients without CLL. That's very important because there are some treatments that have been identified for patients with diffuse large cell lymphoma that can be quite effective in a large proportion of the patients. And those types of therapies may apply to that particular subgroup. So the ones that are not clonally related may be considered more like diffuse large cell lymphoma and less like a variant that's occurring out of CLL. And I think it's very important to look at these genetic changes and try to identify what factors are associated with Richter's transformation that develop out of the leukemic cell clone. Because if you can, then they represent novel areas for further investigation. So we can try to go down to signaling pathways and find out how this Richter's transformation, I'm confident, we'll be able to try and dissect this and discern pathways that we can interfere with in an intelligent way, not just throwing the kitchen sink at these patients and hoping for a better outcome. No, we have to be more intelligent and turn, again, a bad prognosis disease into something that we can treat very readily, and then it becomes just a challenge, but not a life-threatening one. So I think all this is very exciting, what's being discussed at the ASH. And I must say, a lot of things that are being discussed, it's very gratifying to see, because it's a long-held conviction of mine is that if you get the leukemic cells from patients and you can analyze them before treatment or during treatment or after treatment, we can really try to find out how this thing works, how the leukemic cell actually functions, how it may develop, and what makes it progress and become resistant to therapy. And this has been phenomenal because now we have agents which are targeting these pathways rather than just doing a whole cell dump on any cell that divides. We know that the outcome of that is not very, very good. And I hope that further work, I've been spending a lot of time on ROR1 because I have to focus, but ROR1 is very interesting from my standpoint because it represents a developmental protein. Namely, we all have ROR1. It's highly conserved. This protein, if you look at it, has a sequence identity with the mouse ROR1, 97% identical at the protein level. And there's even evidence, you have fruit flies that have a ROR1 protein, and they have it highly conserved in certain regions. It's phenomenal how well conserved it. That means it's important, okay? Why is it important? Well, we know now that the ROR1 is used in early development, namely in embryogenesis. If we think about the miraculous thing that happens with embryogenesis, it's phenomenal. It always blows me away to think about one cell dividing, forming a blastocyst, and then having cells migrate to parts of the blastocyst and form new organs. And then out comes a developed embryo to, it's just amazing. And this is all a choreographed dance. And it has to be some of the proteins that are involved in this migration of cell trafficking. And ROR1 is a participant in that dance. So if you have loss of ROR1 or mutation in ROR1, you get pretty severe developmental defects that result in the problem in developing your organs. And so we know it plays an important role in organogenesis. Now, when the embryo matures and comes to term, it doesn't need to make new organs. So it shuts that pathway down. And it's come into my consideration of what cancer is all about. Cancer is probably a replay of our early developmental programs in embryogenesis gone amok. Instead of keeping it shut down, we've regenerated them, perhaps in a cellular form of quest for immortality. And the development of new organs and what have you that we are vital in embryogenesis are something that we see a caricature in cancer. And now we see that ROR1 may be expressed in a variety of cancers, lung cancer, breast cancer, brain cancer. Why is it there? Why isn't it on normal cells? And if we dive down into what it's doing on the cells, we might be able to come up with some pretty clever ways, even for these really terrible tumors that patients face, and do it in a way that strikes at the biology rather than just strikes at anything that moves.